Hello, everyone. Welcome back to another lecture. And in this particular video, we are going to discuss about the structural organization of proteins and primarily the four stages of protein organization, that is the primary, secondary, tertiary, and quaternary. Now, the proteins which are present inside the biological bodies, they are serving a variety of functions. For example, they are acting as enzymes for carrying out the biological reactions. They are also acting as certain hormones. They are the precursor molecules for several biomolecules. So without proteins, there are a lot of things that will be halted. Now, the functions of the proteins, they are corresponding to the structure of the protein. The structure of the protein is responsible for closely deciding that what function it's going to exhibit inside the biological body. Now, fundamentally, all the proteins, they are made up of the amino acids, and all these amino acids, they are linked to each other through peptide bonds. For example, if this is the amino acid number 1 and this is amino acid number 2, both of these will be connected to each other via CONH bond, and this CONH bond is nothing but the peptide bond. So all these amino acids, they link together to form long chains of peptides, long chains of proteins. Now, after they have made long chains, there is the next level of structural organization that decide that what kind of shape it is going to acquire. And that is what we have to look into this lecture. The active form of the protein, the active structure of the protein, which is of utmost importance, is the tertiary structure. That is the three-dimensional structure. Now, three-dimensional structure of the protein is formed by the coiling and folding of this peptide chain. And there are several different peptide chains that can connect to each other to form the next level of organization, that is the quaternary structure, which we will also discuss in this lecture. Now, the three-dimensional structure of the protein, that is the active conformation, or we also call it as the native conformation. Why do we call it as native conformation or the active conformation? Because the tertiary structure of the protein is the one that is exhibiting its function inside the biological body. For example, if there is an enzyme, you know that enzymes are proteins. If an enzyme has to carry out its function, it has to be in its three-dimensional conformation. Only then it will act and exhibit its function. The three-dimensional structure, also known as the conformation of the protein, has a unique structure corresponding to each protein. So every protein will have a unique three-dimensional conformation. Now, the biological functions, they are dependent on the conformation, as we just discussed. Any change in the conformation will lead to the loss of function. So the protein has to be in its conformation, in its active conformation, and if there will be any change in that, the function of that particular protein will be lost. And the conformation depends on the sequence of the amino acids, because you know that the proteins are made up of amino acids, and when these amino acid chains, they are going to coil up, the kind of bonding they are going to form at different places of the chain, it is going to be dependent on the sequence also. Now let us see that what are the different types of bonds that are present inside the active conformation of the protein, which is responsible for making the protein structure active, which is keeping the structure intact. So there are two types of bonds that are present. First are the covalent bonds, which are the stronger bonds, and the second is the non-covalent bonds, which are the weak bonds. Talking about the covalent or the strong bonds, these include peptide bonds and the disulfide bonds. If you can see here, this is the peptide bond. So this is the amino acid number 1, amino acid number 2, and amino group of one amino acid is making a bond with the carboxyl group of the second amino acid and forming a peptide bond. So this kind of bond is existing in the protein structure. How? When two amino acids are combining to each other. So this bond is present there. Next is disulfide bond. Now, disulfide bonds, if you look here in this figure, This is basically forming between the sulfur containing amino acids, particularly cysteine and methionine. So you know that the cysteine and methionine, they are having a sulfur group present in their structure. So these bonds, the disulfide bonds, are forming between the cysteines present at different locations of the peptide chain. The second category is the non-covalent bond, and non-covalent bonds are weaker than the covalent bonds. And these contribute to the stability of the protein structure. The different kinds of non-covalent bonds present inside the protein structure are hydrogen bonds, particularly which are forming between the OH hydroxyl group containing amino acids, such as tyrosine. So here you can see this kind of hydrogen bond can be formed. Then we have electrostatic bonds, and we have hydrophobic bonds. So these are involving the charges. So the structure of the protein, it has been divided into 4 groups, 4 levels. First is the primary structure. Second is the secondary structure. We have tertiary structure, which I was talking about, the native conformation. And the fourth is the quaternary structure. Primary structure is the amino acid sequence of its polypeptide chain. For example, let's suppose that this is the wire, right? Let's suppose that this is the primary structure of the protein, which means that it is the linear structure. The amino acids, they are connected to each other and they are forming a straight chain. So this is the primary structure. Knowledge of the primary structure is important to understand the functions. Secondary structure means when there are certain kinds of specialized foldings happening inside this chain. For example, let's suppose that this linear chain starts to fold up from here to form some kind of structure, which we will discuss in the coming slides. Or maybe it starts to fold up from here. So these kind of secondary structures start to form due to the interactions between the amino acids present in this chain. The tertiary structure is the 3-dimensional conformation when all the secondary structures join together and there are different forces present between the amino acids present on the different sides of this peptide chain, and hence they finally form a 3-dimensional structure. Let's suppose that this is the 3-dimensional structure now, and this will be having a function. This will be the active conformation. And quaternary structure is when the tertiary structures, let's suppose that this is the tertiary structure of one polypeptide chain, and similarly there is one more tertiary structure of different polypeptide chain, and they interact with each other, and that is where the quaternary structure is formed. I hope I was able to make a little bit better understanding using this example. Now let us see all the structures one by one. Talking about the primary structure, as I just mentioned, that it is the sequence of the amino acid in the polypeptide chain. So primary structure gives us an idea that what sequence of the amino acids is present in that chain. We will understand that, okay, this is the amino acid number 1, next it is bonded to this particular amino acid and so on. For example, if you see here, you can see that this is a polypeptide chain where we have glycine, isoleucine, valine, and so on. So this is a primary structure. And this is the most fundamental level of the structural organization. And each of the chain is having N terminus and a C terminus. What do you mean by N terminus? You know that the amino acids, they are having two groups. That one is amino group and one is carboxylic acid group. So on one end of the chain where the amino group is exposed, where the amino group is free, that is known as the N terminus. And the other side where the carboxylic end is exposed, that is the C terminus. The higher levels of the structural organization, they depend on this primary structure only. And any change in the amino acid sequence will lead to the alteration in the kind of folding happening in the higher level of organization, and hence the structure can also be affected. That is the tertiary structure. Talking about the secondary structures I was telling you, this is the next higher level of organization after the primary structure. The polypeptide chain, it takes twists and turns on different areas of the polypeptide chain to form the secondary structures. And the most common and important secondary structures involve alpha helices, beta pleated sheets, and beta bends. Now, what is an alpha helix? Alpha helix is when the polypeptide chain coils up to form a helical structure by the formation of hydrogen bonds between the peptide linkages. For example, this area. So it will start to coil up. So this kind of structure is known as the secondary structure. The beta pleated sheets, on the other hand, sides, they are the portion of the same peptide chain or the different peptide chains running side by side, and they are joined together by the hydrogen bonds. So you can see here this kind of structure. So when one polypeptide, like let's say there are two segments of the peptide chain, and one segment is running in this direction and the other is running in this direction. So this kind of parallel kind of structure, that will be known as the beta pleated sheet. Talking about the tertiary structure. Now, what is a tertiary structure? Tertiary structure is the polypeptide chain folded in complex ways, as I showed you in the example of that wire. So when the secondary structures are formed and now there is a full conformation formed where different amino acids on different sides have made the respective interactions with each other, that becomes a tertiary structure. And this folding produces different types of secondary structures in different regions of the chains that we just discussed. The bonds present are disulfide bonds, hydrogen bonds, electrostatic bonds, and the hydrophobic bonds. And some of the amino acid residues which are distant from each other in the polypeptide chain, they are brought together. For example, let's say that amino acid is present here on this spot and the other amino acid is present on this spot. Due to the folding, these two amino acids are coming in close proximity to each other, and there is a bond formation, and that is also contributing to the formation of this tertiary structure. And due to the tertiary structure formation, some of the amino acids, they are buried deep inside the molecule, and some of them will be exposed on the outside. Now, how is this happening? So the hydrophobic interactions, they will be towards the interior, whereas the amino acids which are having hydrophilic character, they will be probably on the outside of the structure. So that is why the composition of the amino acids in the peptide chain is important, because due to that only, due to their hydrophilic and hydrophobic characteristics and the kind of molecules or atoms like sulfur or oxygen, whatever they are having inside them, that is going to govern that what amino acids are going to contribute towards the interior of the structure and what amino acids are going to contribute towards the outside of the structure. Next is the quaternary structure. What is quaternary structure? Quaternary structure are the structures that are made up of two or more polypeptide chain. And the most important example in this is hemoglobin. Now, hemoglobin is made up of four chains, four individual peptide chains. They are different peptide chains. They are forming their respective tertiary structures. And then these four tertiary structures are bonding to each other to form this quaternary structure of hemoglobin. So in case of hemoglobin, there are two alpha chains and two beta chains, and these are interacting with each other. Now, the subunits which are present in the quaternary structure, they can be similar or they can be dissimilar. Then there is a possibility that there are two copies of the same peptide chain in the three-dimensional conformation forming a bond with each other to form the quaternary structure, or maybe they are dissimilar to each other. This peptide is different, this peptide is different. They are forming their respective tertiary structures and making a bond with each other. And each chain in this quaternary structure is known as a protomer or a subunit. So this was the level of organization inside the proteins, and that's it. In the next lecture, we will talk about enzymes.